Hello and welcome to my talk. My name is Edmund Basham. I'm a PhD student at the University of Florida studying ecology, and today I'm going to talk to you about this cool frog, the yellow-bellied poison frog, known as Andinobatis vulgaritus. So the title of my talk is The Chemical Defenses That Shift with the Seasonal Vertical Migration of a Panamanian Poison Frog, which is our yellow-bellied poison frog. This study was published in Biotropica last year, and please go and have a look at it. And if you want to cite it, please go ahead. I would love it if you could read it, and if you have any questions, send me an email. So this title really touches on a lot of different topics. So the toxicology of the frog, what kind of alkaloids, the poisons, the toxins that it has in its skin, its movement ecology, so how the frog changes its habitat use across different seasons and it actually moves from the ground to the canopy across seasons, its diet, the consumption of ants and mites is what drives the toxicology of this frog and Dendrobates in general. And also thinking about climate and how climate influences its vertical position. So across different seasons, the wet season, they go up into the canopy, and the dry season, they come down to the ground. And that causes a change in the potential arthropods that they have to eat, and that might change their poisons. So the objectives of my talk would be to give you a background on the system, present my research objectives, run down some of the methods, the results, and the conclusions. So the background is that dendrobatid poison frogs, you've seen them on nature documentaries, the incredible, colorful little frogs that live in Central and South America. They sequester toxic alkaloids from arthropods. They don't actually produce the poisons themselves. And different alkaloids are found in different arthropods. Interestingly, especially in tropical forests, you get incredible significant shifts in arthropod communities across even small scales. And talking about across the vertical height of the forest of one tree, from the ground to the canopy, you could have completely different species of arthropods occurring. And many tropical frogs, seen in Basham and Sheffer's 2020, one of my other papers, we showed that frogs migrate vertically from ground to the canopy with seasonal patterns of rainfall, including this cool poison frog. So one of my papers that's currently in review at Ecologia, we showed that there's a distinct migration between the terrestrial habitat in the dry season, arboreal habitat in the wet season, and they actually breed up in the canopy using phytotomata, which are plants which have water in them, and they deposit their tadpoles in these tiny plants to nurse them. And so this is a plot from that paper showing the height distribution of individuals in each season. And these were vertical surveys that I actually climbed up into the tree, 40 meters, vertical surveys, collected the frogs, and this is the data just showing that dry season, they're nearly all of them found on the ground. And in the wet season, nearly all of them found above ground, and lots right up into the canopies. There's 20 to 30 meters up, really high up in the canopy. And so that comes with it a change in microhabitat and a change in the different plants that are available for different arthropods, herbivorous arthropods, and then the predators of those arthropods. So you get a complete shift in potential dietary items for these frogs. So my objectives were to measure the shifts in alkaloid profiles of this frog across seasons, which coincides with their vertical shift in habitat, provide the first description of alkaloids sequestered by this frog, because they've never been assayed before, and make predictions on the potential impacts of climate change on the defensive capability of this species, which is an additional really cool thing that we can start to talk about. So I'm just going to quickly run through my methods. This is a quick talk, so let's not get stuck anywhere. I did my study in Panama, in central Panama. I took frogs from 6 different trees. Now, this was part of a bigger study, and I collected loads of frogs from loads of different trees, but I selected a particular set so that I could have the same number of frogs from the same tree across different seasons so that I could ensure replicability of my samples across space. And so this is an example of one of these trees, an EMPSEB, which is Anacardium excelsum, wild cashew tree. And I found that these frogs only seem to live on these massive, massive trees in this forest. And so each one of those trees I showed in the last one is the same species of tree. And that's really useful actually, because it means that we can reduce the variation in arthropods that we might expect to occur between tree species because of different epiphytes and different microhabitats that those arthropods would live on. So we've got frogs living up in these absolutely monstrous trees, super cool. I took 13 frogs from each season and I extracted the alkaloids from these frogs. I used a TAS, which is essentially a tiny little electric stimulator, to buzz the frog, and it just makes it tense up ever so slightly and pushes its poison out, and then you can collect it with an absorbent piece of paper. It does not kill the frog. I really didn't want to kill the frog, which is what most poison studies will have to do, is they extract the skin. I didn't want to do that, so I just gave him a little shock and took the poison. We can then run that through gas chromatography, mass spectrometry, and other kind of sci-fi machines to create curves and weights of the different molecules and blasting them with electrons. And from that, you can figure out, using a nicotine standard and other methods, the actual quantity of individual alkaloid types in the frog, and different arthropods from other studies that have been shown. Different arthropods produce specific structures of alkaloids. And so when you can see a specific structure in a frog, you know that it had come from a specific arthropod. And you can actually get it to ants or mites or beetles or millipedes, that kind of level. So we found that this is our quantity on the left and richness of alkaloids on the right. So the actual amount and then the number of types of alkaloids that came from ants and mites, which were our two sources, essentially. There were no other sources that we found. And interestingly, we've got more alkaloid coming from mites than ants. And that's actually really funny, because most studies will say these species, these tiny frogs, are ant specialists, they only eat ants. And that's probably where they get all their poisons. Well, just so happens that some of them don't. They actually eat a lot of mites. And so it may be something to do with the frog's size. It's a tiny little frog. It's only 14 millimeters long. It's a tiny, tiny frog. And so it's eating these tiny little mites on these big trees. Such a cool system. So we actually found that they had less alkaloids in the dry season. So these frogs that we took from the dry season had only an average of 84 micrograms per gram of frog versus 139 in the wet season. Now, there was a lot of variation between our individuals, as you might expect. Some frogs have loads of poison, some frogs don't have very much. And that's why our p-value is not absolutely huge, but it's still significant. And, you know, there's definitely a difference in the alkaloid quantity between frogs, whereas the richness didn't actually change. And this is probably because that they can sequester, they can keep poisons for quite a long time. So over time, they'll have a decrease in abundance, but they'll always still have a little bit left to see in the curve. So you'll get the richness, but it might not necessarily have the abundance after time. So we can also do an MDS analysis, a visualization of the compositional differences of alkaloids between frogs. And so each point is a frog taken from the red, dry season, the wet, blue. And the further away the points are, the more dissimilar are their poisons. And we can see that there is a clear grouping of red here and blue here. There's a separation in the composition between seasons. So they are eating different arthropods and they're getting different alkaloids across the seasons, which is really interesting. And this is just a representation of what we previously saw with the height distribution of the frogs, just to match up to show that in the canopy, in the wet season, they're up here, and in the dry season, they're down here on the ground. So to bring these conclusions together, Fulguritis primarily sequesters its defensive alkaloids from mites, but ants are also very important. The composition and abundance of alkaloids differs between frogs using seasonal, ground, and canopy vertical habitats. And then how can we link this to climate? Climate change is predicted to increase the length and severity of dry seasons across tropical regions, including what look like, super wet tropical forests can actually then have pronounced dry seasons. And so if this frog is moving down to the ground in the dry season, it might be forced down to the ground for longer. And therefore, if following our results, it may end up with a gradual decrease in the amount of poison over time. And this will decrease its defensive potential and potentially its fitness. Now, this is just a theory, and it'd be really cool to test this in a few years, but it's a really interesting prediction that we can make of this data. For more information, please have a look at my website and have a look at this cool research video that I'm linking here, where I just talk about my general research in Panama. And please go and have a look. I can't stop without saying thank you to all of the lovely people that helped me. The team in Panama, these awesome guys helped me sampling in the forest, as well as numerous other people along the way. Dr. Ralph Saporito, who was instrumental with the alkaloid analysis, my various funders, and of course my academic lab. Dr. Brett Sheffers, who's my PI, is a big help for this project. So thank you, everyone. Thank you very much for listening, and feel free to ask some questions. I'm here to answer them. Thanks very much.